To the naked eye, a forest might look resilient, but hidden inside every leaf within the double helix of DNA, a countdown is happening. As our climate shifts faster than at any point in human history, plants are facing a survival gap. This is the story of genomic vulnerability, the science of predicting which species will adapt and which are standing on a genetic precipice. In the past, we judged a plant's risk by its habitat. If the forest got too hot, we assumed the plant was in trouble. But today we look deeper. Genomic vulnerability isn't just about where a plant lives, it's about its genomic offset. Think of it like a biological mismatch. Scientists use machine learning to map the genetic traits a plant needs to survive today. Then they project those needs into the year 2050 or 2080. The offset is the distance between the genes the plant has and the genes it needs to survive that future climate. A high offset means a high risk of extinction. Essentially, the plant's evolutionary library doesn't have the right books to solve the problems of tomorrow. Why are some plants more vulnerable than others? There are three major red flags. First, low genetic diversity. If a population is small, it has fewer genetic options for natural selection to choose from. Second, the time factor. Trees like the Great Basin bristlecone pine can live for thousands of years, but their long lives mean they reproduce slowly. They can't evolve as fast as a weed that produces new seeds every few months. Third, habitat traps. A plant living on a mountain peak or a small island has nowhere to go. If its genome isn't ready for the heat, it can't just walk north. It's not just about what's missing. It's also about what's weighing them down. This is called genomic load. Over centuries, plants accumulate tiny, harmful mutations. In a stable world, these don't matter much. But under the stress of a 2026 heat wave or a decade-long drought, these genetic glitches can become fatal, making a species crumble under pressure. But it's not all bad news. By identifying vulnerability, we can intervene. Assisted migration. We can move seeds from pre-adapted populations, plants already living in hot climates, to help boost the gene pool of northern forests. Genomic editing. Tools like CRISPR allow us to fix specific vulnerabilities in vital crops like rice or coffee. Herbarium data. By sequencing DNA from 100-year-old dried plants at institutions like the NYBG Herbarium, we can see how plants adapted to past changes to predict their future. Nature is incredibly adaptable, but we have accelerated the clock. By understanding genomic vulnerability, we aren't just watching species disappear. We're gaining the blueprints to help them fight back. The future of our forests is written in their DNA. It's up to us to learn how to read it.